A very good afternoon to all. On behalf of President Dr. Vandana Prasad Ma'am, Director BSIP of the Palaeobotanical Society, I would like to welcome you all for the Palaeobotanical Society second lecture. We thank the speaker of the day, Professor Kevin McCartney, for accepting our invitation to deliver today's online lecture on the topic Silicoflagellates, Skeletal Morphology and Evolution. Dr. Professor Kevin here is here with us in the institute on the Boy Scout Fulbright Fellowship, and he is working with Dr. Amit Ghosh. This society shall now aim to organize lecture regularly in both online and offline mode on various topics of paleobotanical and allied aspects. Today's lecture by Professor Kevin McCartney is the second in the series of the lecture to be organized this year. We shall keep you updated by email all the time. We shall be recording today's event, and YouTube link for the same will be shared to all. Before we start the program, I would like to ask our President and the Director of the Institute, Dr. Vandana Prasad Ma'am, to welcome Professor Kevin with the bouquet.
Oh my God! Oh, these flowers. Just a moment. Thank you. Thank you. I will give this to my friend.
We can keep it there.
You post it in your blog, I think.
Yeah.
Send me a picture. I'll put it in my blog.
Now I would like to ask Dr. Neha Agrawal to introduce today's speaker, Professor Kevin McCartney. Dr. Neha, please.
Thank you, sir. Very good afternoon to all of you. I'm going to introduce today's speaker, Professor Kevin. Professor Kevin McCartney has pursued his research in Florida State University in 1988. His topic of research was modeling silicoflagellate skeletal morphology. Presently, he is working as a professor in Department of Geology, University of Maine at Presley. Besides it, he has earned various academic and community positions. Few of them are Director Founder, Micropaleontology Undergraduate Research Lab since 1992 to present. Director Founder, Northern Maine Museum of Science since 1996 to present. Maine Solar System Model, Larval, Western Hemisphere, built 1999 to 2003. Educational Outreach Participant, New Horizons, Spacecraft to Pluto, 2006 to 2015. Aroostook County Representative, Down East Friends of Folk Art, 1998 to 2008. Aroostook County Representative, 1997 to 2006. Board Member Director, Northern Maine Agricultural Fair, 2002 to present. Co-founder, Historical Pavilion, Northern Maine Agricultural Fair, 2000 to 2006. Chair Founder, Musical Festival, Northern Maine Agricultural Fair since 2005 to 2015. Vice President, Northern Maine Agricultural Fair, 2008 to 2010, and many more. He has been awarded Fulbright and Fujitsu Fellowship at Poland. He has published near about 60 research articles in various national and international journals. Some of them are Journal of Nanoplankton Research, Geomarine Lattice, Journal of Micropaleontology, Marine Micropaleontology, Paleogeography, Paleoclimatology, Paleoecology. Kevin studies the silicoflagellate group of single-celled algae with siliceous skeletons that are common in modern ocean and has as fossils since lower Cretaceous. He has studied this subject for almost 40 years. Today, he is going to deliver a talk on the topic Silicoflagellates, Skeletal Morphology and Evolution. With this short CV, I invite Professor Kevin to deliver his talk.
Should I start or wait till we hit 4:30?
Please. Already 4:30.
Okay.
Yes.
Okay, well, let me then start talking about silicoflagellates. As you've heard, I've been focused on this particular subject for 40 years. And I know we have, first of all, can everybody hear me? Can somebody out there say they can or can't hear me? Audible to everybody. Okay?
Yeah.
So, let me start with the larger picture. We'll hone down to the subject of silicoflagellates, and we can talk about the bigger picture again. If you look at the modern record in the oceans and in the fossil record as well, focusing on skeletons of carbonate or siliceous, there are 4 really, really important groups in terms of this in the modern oceans. And these are, on your left side are those 2 very important groups made of carbonate skeletal material, and on the right, siliceous. The top are the autotrophic, the plants, the bottom are the heterotrophic, the animals. And each of these 4 groups has its specialized scientific organizations. Most of these have specialized journals and so forth. And the importance of these cannot be understated. The rule of thumb is that at each trophic level, only 10% of the biomass gets to the next trophic level. The bottom trophic level is the heterotrophs, the plants. That makes up something like 90% of biomass. And the second group, which is the second row here, would only at most be 10%. And just that there is no paper I know that tries to quantify the exact proportions of these in the global biomass. But I'm going to say, my guess, having seen a lot of things in my life, I think the coccoliths are maybe 20% of Earth biomass, a very important group. Diatoms, probably very close to that as well. The foraminifera and radiolarians probably represent close to half of the animal portion of the biosphere. We're again fixated on the continental areas, but the oceans are much bigger and much deeper. So there are the 4 really important groups, probably representing close to half, maybe, of global biomass, just a guess. There is a 5th element. There is a 5th, what's considered to be an important group in the oceans of carbonate or siliceous skeletal material. This one is, though, a significant series of rungs below the 4 that we've talked already. These are maybe a 10th to a 20th as important as the groups that I have mentioned previously. And as such, it does not have a special society, it does not have a special journal, it does not have, whereas the other groups we had have hundreds, if not thousands, of scientists studying that particular group. For silicoflagellates, more or less, I am it. So let's talk about these. They are an extremely interesting group. One thing, I'll just go back to my previous image here. The group is very simple, I think very attractive skeletal designs. These skeletons are rigid latticework skeletons. That kind of structural context is very unusual in the organic world. Actually, basically 3 groups in the organic realm have rigid 3-dimensional latticeworks, and all of them are siliceous, siliceous single-celled. And the reason probably is because the ocean is undersaturated in silica. The seashells can be continuous shelled because they don't have to worry too much about getting the calcite for that. So the 3 groups are the silicoflagellates, the abridians, and the radiolarians. I'll go back again here. The abridians and radiolarians have internal skeletal elements. The silicoflagellates do not have internal skeletal elements, and they also are different from the other 2 in that the skeletal elements themselves are hollow. So they're very much saving on skeletal material here. There is considerable diversity in shapes and size. The pictures on the top here are from the early Eocene Arctic Ocean. We are on the far left see a skeleton, and we're looking down at the top, what we call the apical structure. The middle one, we're looking up through the basal ring to the bottom of the apical structure. This on the left, we can go to the image below, looking down the apical structure is called apical view, and looking up through the basal ring is called abapical view. And as I said, the group is it's remarkable, okay? This group, I think, has greater variability than any other fossil-bearing group of organisms on the planet. The variability comes in several ways. It is considerable. We'll talk about two basic types of the usual, within a species, you have variability. We're all different heights and all the other stuff. Although if you're looking at a group of six-sided silicoflagellate skeletons, a species that you recognize as generally being six-sided, you always will see some five-sided, some seven-sided, some other variation. And let's just take a little look at some of that in the context of the Stephanopsis crux. Back in the 1940s, 1950s, there were several scientists, Fringuelli, de Flandre, who were looking at silicoflagellates, and they emphasized the variability. Whole plates showing the variability within a particular taxon. But in the 1960s, with deep ocean drilling, people were talking about biostratigraphic zonations, and people got into this variability of silicoflagellates and looked at every single shape, giving it a scientific name, which rather quickly got elevated to species status. And then other people would get in with very minimal silicoflagellate experience would say, hey, that's sort of different. I'll make a new species out of that. And so we wound up with an extraordinarily split taxonomy. This particular, can you turn off the things that beep or? No. Okay. So this is a range of pictures, come from a paper, a typical paper from that realm. You are looking at all these photographs come within 10 meters of core. The one at the upper left is recognized as being a variant, okay? But the other three are all have separate species names. Actually, the species names are in two genera, okay? And for me, my experience tells me that this is all variation within a species. So there's different points of view within this group. And what I've been doing, and this is from a paper that I've just published in Marine Micropaleontology, looking at a variety of cores to determine some of the evolutionary relationships within the group over the Cenozoic. What I am increasingly doing now is I'm simply, within a species, I will name some of the basic variants. I'm not going to worry about the scientific names. Ones that have two windows, ones that have five sides, one that has an apical ring or apical structure that does not have a ring. This is for Stephanopus speculum. There's five-sided, there's seven-sided, there's two windows, there's more windows, and so forth. Quantifying them simply so people can see what that range of variability is within the species. So as I said, for silicoflagellate variability, you have this considerable variability within individual taxa. But there's something else, and this is something that I don't know if there is in any other group that I can think of, at least in the manner that it occurs in the silicoflagellates, and that is what's called plexed. These are episodes of time, probably limited geographically, where you have extreme variability. And this is the first plexus episode I recognized, and I've recognized about 12 of these episodes, in different genera. Cretaceous to Miocene. This is the mother taxon, a six-sided with a six-sided ring, and yet you don't see any of that morphology here. Instead, you see all these other kinds of things, and these skeletons are far more abundant than they normally are. They are all over the place. They're dominating, and they're dominating for some number of tens of thousands of years. They're they're saying loud and clear, hey, something's really going on. You wouldn't be able to recognize this in the other taxa except by their mere presence or absence, but here you get all these different shapes that are telling you that things are interesting during that time. So, one interesting thing about the silicoflagellates is that there are definite rules in the way that these skeletons are formed. And this is a series of rules. They're domal design, near hemispherical, skeletal components, connect with triple junctions, and so forth. Let's just take a look at the first two of these. So, silicoflagellates have a hemispherical shape. We have the apical structure centered over the middle of this, around it, no internal elements. If you want to try to imagine this, and perhaps my two-dimensional drawing is think of the camper's dome, the skeletal elements, the tent itself, at all. That is basically what a silicoflagellate skeleton does. And as I said, all these skeletal components are meeting in triple junctions, so the general structure is something like a geodesic dome. So when I was working on my PhD, I just puzzling with why these rules, why these simple morphologies, what's going on here. I was talking to one of my committee members about the subject that I had chosen, and I said, you know, they're sort of like soap bubbles. And my faculty member said, do you know much about soap bubbles? And I said, I don't know anything about soap bubbles. And he had an article published in Scientific American about the mathematics of soap bubble design. And this is an illustration from that paper. It's talking about minimization structures. Now, if I were to draw, you know, make 4 points, the corners of a square, put them on a map, and say, okay, now we have a pipeline that's going to connect all these points. You would think and I want those pipelines to be of minimal length because there's building material and so forth. You might get a line would look like this, but that is not the case. Actually, there'll be a bridge here where it shares the length there as it distributes the oil or whatever to all the 4 points. This changed my life because the patterns you see here for connecting 4 points, connecting 5 points, connecting 6 points are all found and very commonly found in silicoflagellates angles. Okay, so the solution to my problem is to be found in soap bubble mathematics. And I'm not a mathematician, and even if I was a mathematician, the mathematics here is very specialized. Walking around the math department, talking to people, and eventually I got a couple people saying, well, there's nobody here in the mathematics department that can help you here, but you might talk to Dave Loper in the Geophysical Fluid Dynamics Institute. And I came in there unannounced and announced, hi, my name is Kevin. I am got a paleontological problem with a mathematical solution. And he said, okay, let's work on that. And what we did, and he's the one doing mathematics, but rules and the shapes. And we are, for a particular shape, what is the minimal optimal design? What the exact assumptions were, and that took me a year or two. And there would also be variables in here done so that once we get that minimal design, I can actually determine the shape of the skeleton. And what we found out is that skeletons enclose a spherical shape, constrained available silica, and what they're doing is they're minimizing surface area for internal volume. And with that done, 2 or 3 years later, I can test this. I actually measured several thousand specimens, various shapes, and what I'm doing is flat line, and I am digitizing the points around junctions around the basal ring. I actually put a a device to measure the amount of rotation of my fine adjustment knobs. And from that, these lengths, and I can put these in a computer program and find out what these are broken into nice little facets and what the internal volume is. And those numbers came to very close to what the mathematics was predicting. Now, all that said, then I can tell you that the six-sided shape with the apical ring here, that is, of the two shapes that I show here, that is the one that better reduces surface area to volume. Okay, then why do we have these simpler designs? And here is the pattern within the world today. Again, this now known as Stephanoka is the dominant design in high latitudes. The simpler design, however, is dominant in low latitudes. And by the way, in the evolution of Silcoplagiazoic, the more complex skeletal designs are always in high latitudes, simpler in low latitudes. There's been a gradual change in the players in the game, but the relationships stay the same. And what's going on here? It turns out that if you constrain your formula to wait for the minimization of apical surface area with some weighting for silica conservation, then you find that where the area is absolutely essential, you get these things of these two places. Where silica conservation becomes more important, then you get the dictyoka. And that is the situation that we have here. Simpler designs where skeletal material minimization or reduction is more essential. More complicated designs where the minimization of surface area is essential. Okay, so these are optimization structures. They minimize surface area for internal volume. So, why are these skeletons hemispherical? I mean, come on, look at these again. If you are going to reduce your surface area to an absolute minimum, you're going to come up with a sphere. Euphorians and radiolarians have spheres. Silcoplagiates don't have spheres. They have hemispheres. There's a hemispherical shape. But the reality of it all is that Silcoplagiates don't have spheres. Silcoplagiates have spherical shapes when they need them. And when they need them is during mitotic division. It happens when they are reproducing. They form a skeleton at the basal ring, another skeleton attached to the basal ring. They do their business, then they separate. And by the way, soon after separation, these organisms get rid of the skeletons. They only need the skeletons during mitotic division. Interesting. So silcoplagial skeletons are indeed spherical. This business of double skeletons, I had no particular interest in. I had no particular interest in double skeletons. I had no particular interest. Turn that off, or we're having a technical issue here. Sorry about this. So I have no particular interest in double skeletons because they split, and I don't see them. 30 years worth of looking at, or 25 years worth of looking at silcoplagiates, I'd never seen a double skeleton. But I found one in the Cretaceous, and it was odd. And it made me start asking about double skeletons. And so you look through the literature, and I found out there was almost no literature on silcoplagiate double skeletons. This is a list of every illustrated double skeleton of a silcoflagellate in 125 years of scientific literature. This is it. So I need to know more. Fortunately, I have a network of scientists that I work with, and they have various photos. We started pulling it all together, and we went up with a paper on the double skeletons of the three genera in modern waters. This is for Stephanoka again. Let's look at that in the fossil record. Now, the image you see on your right, this is that first image that I saw in the Cretaceous. And it's a double skeleton. I'm excited, but it's not a double skeleton like any double skeleton that I've seen in any of those pictures that I showed you previously, because these two skeletons seem to be rotated with respect to one another. Actually, to be more specific, they're flipped with respect to one another. And that's off, because all modern silicoflagellates, and what few that were known in the fossil record, are corner aligned. So I've got what I call a Star of David configuration on one side, the corner line on the other. Here are some of the skeletons that we found over the course of a couple of years of frenzied activity. The three you see in the middle left upper end, these are corner aligned. These are all Eocene except the one on the upper right, which is Paleocene. So we're looking at lower Paleogene. So the ones at the bottom are Star of David. Here are some more pictures. Again, the two at the top are fossil, the three at the bottom are modern. The one in the lower right is, I think, the best photograph of a silicoflagellate of any kind ever taken. We have, this is a somewhat elongated three-sided shape, and it's a double skeleton. The other one is flipped, 180 degrees. And the other thing that's interesting is we've got these pikes. Now, these pikes serve different purposes in different silicoflagellates, but for this group, these pikes seem to be actually touching that, that soap bubbled area in the middle of this opening here, sort of like a tent pole would be. And I think these are actually helping to stabilize these larger openings, again, during mitotic division. And with all that done, why do we have these two groups? And my conclusion is that what we have is we have two very broadly different silicoflagellate groups here. The Star of David, in this early illustration, I have them diverging into Cretaceous. I now think it's soon after the KT boundary, very early evolution within the Paleogene. The Star of David configuration persists until the Oligocene. The corner aligned, which is the novelty after the KT boundary, the corner line configuration diversifies and continues to the present. So this is the Paleogene evolution based on double skeletons. And again, I just published a paper about 2 weeks ago on marine micropaleontology. This is a little bit more elaborate phenogram. And what you see in the lower part here is the Star of David group. And then in the upper part, we see the corner aligned double skeletons. Okay, so why does all this matter to us? And at this point, I'm going to go back to the larger scene. I don't want to get too mired in the nitty-gritty of silicoflagellates. I certainly can go on for quite some time that way, but silicoflagellates is a minor group, and even in the micropaleontological realm, it's not something that people spend a lot of time talking about. Why does this matter? And why does this matter not just to micropaleontologists, why does this matter to all of us? And let us remind ourselves the four basic groups here that we started with. Again, the top is carbonate, the left is carbonate, the right is siliceous, the top is plants, the bottom is animals. One of the other things I worked on when I was a PhD student, also with Dave Loper, he was in fancy mathematical modeling of mammal plumes going through the mammal, and we played with the idea that those could be responsible for mass extinction events, and it's still something that's argued. So let's talk a little bit about that KT boundary. In the KT boundary, this left column here essentially ceased to exist. There's maybe 1 to 3 species of Coccoliths and Foraminifera that barely managed to survive, and then everything that we have had since then is descendants of those very, very few species. Understanding that in the Cretaceous, the carbon, this was the stuff that made chocolates of Dover. These things were very important, and the KT boundary wiped them out, essentially. And why did that happen? And people love to blame it on the meteorite itself, but it may well have been something chemical within the water itself. And let's also remind ourselves of the relative importance of the things in the ocean. I am an undergraduate course, a teacher. I always see, whenever I teach oceanography, I get somebody coming to me and say, "Oh, I'm so excited to take this course because I love to learn about whales." And I say, why would we spend any time on something as ridiculously trivial as whales? And their jaw drops and we move on from there. Let's remind ourselves what's truly important in the oceans is this represents maybe something like 90% of Earth's biomass, and it represents maybe 90% of that remaining 10% in general terms. This is another guess of mine here, these are outrageously important. We're going into climate change, global warming, and we're in a world where everybody's concerned about atmospheric CO2 and they're concerned about glaciers melting and sea level rise. And that is the least of our concerns. And I'm sure I'm speaking to an audience that knows this very well, but it's worth reflecting in any case. Because the CO2 is not staying into the oceans. And I don't really think so points are reached, but this is the heat content. If you look on the list, it's an order of magnitude in a matter of a few decades. And what happens as you warm the water, warm the oceans, you're increasing the amount of dissolved carbon dioxide and you're decreasing the pH. The pH is a logarithmic scale, so it doesn't look like numbers very much, but they really are important. It's called acidification. And again, I'll just go back here. This is a very stylized diagram, but it's always nice. The scientists like to look at data. The red curve here, that's the Mauna Loa atmospheric CO2, and that's interesting enough. But I think more important to us is the dissolved CO2, and even more important there is the pH, which is going down. Yes. And this is I don't know if it's famous here. It seems to be shown in every talk that I see in the United States. So it seems to be a famous cover of Oceanography magazine. And here's somebody who did a study. They're looking at larval gastropod shells. Then these float in the water column. And this particular species now, at least in some areas, cannot create naked shells anymore. They're dissolving as quickly as the gastropods trying to put these things together. And that's bad news for the gastropods. And that's going to be bad news for calcareous nanoplankton. I'm a member of the Nanoplankton Research Society, and I don't study nanoplankton. I've been to a couple of national meetings. And when you talk to them, you know, they're all scientific and they're hedging their bets and everything else during the talks. But when you talk to them in the evening, over, you know, after the second or third beer, and you ask them what's going to be happening, I've heard estimates that, you know, these things could have a collapse in the next couple of decades. Okay, we could lose that nanoplankton, and if we lose that nanoplankton, we're going to lose a very large portion of the rest. It's going to be the equivalent of the KT boundary without the meteorite. So these coccoliths and everything else that's in that environment become really, really important to us. And we need a lot about these things. And we don't have the people, we don't have the resources, and we're not doing a good job of it. Okay? I'll go ahead, let's just put that silica flagellate up there. I'll just go and tell you what I think. I think in the oceans right now, the silica flagellates are probably going through one of those plexid events. I'm not saying necessarily ocean-wide. In certain environments, I think it's going to be increasing. I think the silica flagellates are the canary in the coal mine. And I've mentioned that to a couple of my colleagues in India, and I have to explain it, so let me go ahead and give this analogy. In the United States, the United States is essentially a coal mining country. And in the old coal mines, the underground coal mines, the one thing that miners are worried about is they're worried about gas coming out of the coal fields, which can asphyxiate you, it can blow up, and all that kind of stuff. Well, their safeguard there is they bring with them a canary in a canary cage. And the canary just chirps away. Even in a coal mine, it chirps away. It's dead. Out of the coal mine, you go in a hurry. The canary, which is more susceptible to gas, having died, lets you know if you don't get out in a hurry, the same thing's going to happen to you. And I think that the silicoflagellates are the canary in the coal mine for our planet, in the sense that because they go very variable with greater abundance during geologically important events, and these events sometimes events that we don't even know things are going really strange in because we haven't seen the additional evidence. We know the silicoflagellates are telling us that things are bad. So I'm telling you that I think these silicoflagellates, if not already, will shortly soon be going through a Plexid episode. And we have nobody watching, okay? And it's not just for silicoflagellates. You know, there's only a limited number of oceanographic ships that are doing their kind of thing. There's no oceanographic ship at a place or otherwise taking samples year to year, season to season, seeing what's happening as the seasons go by. There's nothing like that. There's nobody watching silicoflagellates at all. There's not really much of anybody watching in the open ocean these kinds of things, and there needs to be. It's important, and the time is ticking for us. So this is my favorite photograph of a silicoflagellate. I understand that copies of some of my papers have been passed out. If anybody has any questions about anything that I've been talking about. Thank you. So we must have some questions here around this table and out there in the peanut gallery. Can we turn on so that they can ask questions if they wish to?
I mean, they are on already.
Okay. Hello out there. Someone ask a question. Take a sip of coffee.
Talk is open for the discussion if anyone has any question.
Nice talk. We really enjoyed because we know very little about silicoflagellates. So one question would I like to ask you, that the time of origin of these silicoflagellates, when it happened.
Another story, I was associated with a short lab participant for DSDP leg 93, actually ODP leg 113, and a colleague of mine said, well, I've got some stuff in the early Cretaceous, and he describes those to me, and I said, well, those are sponge spicules. He said, I think you should look at these. And what they were silicoflagellates. They were silicoflagellates that did not have the basal rings. They had very different skeletal designs. But they obeyed all the rules, including the hollow elements and everything else. And I've since had the opportunity to take a look at some stuff from the middle of the late Cretaceous, and just all kinds of strange things. So I spent some time in the Cretaceous. I worked in the Cretaceous. Of course, I found a Cretaceous double skeleton, which gave me the idea of looking at modern double skeletons. And as soon as I looked at that, it answered some taxonomic issues that I think the biologists were having, that I suddenly knew something that they didn't. It's been an interesting time. Cretaceous and Cenozoic, and I've now done the evolution of both those eras. But they go back to as with diatoms themselves, they go back to the late early Cretaceous, but they're so well structured and they're diverse enough that we know that they go back, and there's some scattered evidence for the diatoms that they go back into the Jurassic for sure, and that may be true for silicoflagellates as well. One of the problems is if they're deeply buried, and of course there's very little sediments like that of that age in the current ocean. If they're deeply buried, that all goes to church. So there's there now. Hello?
I'll allow you to ask some questions.
So I was generally interested with the evolution studies that Kevin do, and mostly he has got the double skeleton and the single skeleton. So I want to know that the double skeleton was the earlier form or the single skeleton. So how can you put those two structures in the evolutionary tree? And what was the need of the double skeleton in the latter one?
So the silicoflagellates apparently need to have something like a structured spherically shaped volume in order to do their divisional processes. So that's what the skeleton does. The skeleton creates that shape that they need at that time, and then they split and go off their individual ways. They've been doing that for as long as there have been silicoflagellates, in all likelihood. One of the things of course, I found one double skeleton in Cretaceous. We found a couple more. These very, very strange designs. Trying to explain those strange designs, if you start modeling, what I did was I made a model of one of these double skeletons, and it's just a weird thing. And I have another one, and they fit right together, just like two pieces of puzzle. So even though we haven't seen actually these double skeletons, we can interpret through the modeling that these things are doing this. And they've been doing that probably throughout their history. So it's not a matter that one chicken happened before the egg, as with the chicken and the egg, they both happen at the same time. So with the fossil double skeletons, they're extraordinarily rare. When in fossil double skeletons, I think there's probably about 40 known specimens now. And for in the Star of David group and in the Corner Line group, there are 3 and 4-sided members that you can't tell necessarily which group they belong to, and that makes interpretation or evolution very difficult.
Okay, thanks.
Anybody else? That is a beautiful picture, isn't it?
Very younger people.
Can I ask one? Yeah, I could understand one was they had these corners and the other.
So one of these corners is actually flipped degrees. So this corner is replicated on the opposite corner of the paired skeleton.
Okay. Any reason why is there? Different structures.
Well, there is apparently they were flipped with respect to one another. And the Cretaceous, that group extends to the Oligocene, and in the early Paleocene, a very small 3-sided form developed the corner line. One of the reasons that I think they were rotated originally is the openings couldn't get too large. And they're basically soap bubble designs, and they find in the early Paleocene, there was a very small skeleton which went into the corner line that holds the two skeletons together more efficiently. And because the holes were small, they could get away with that. And in the course of time, the silicoflagellates have developed strategies, including having skeletons with more witnesses to allow them to do that.
So this is not related to any pH or anything in the ocean. These two different structures.
Well, I would say that the change from the Star of David to Corner Line is probably not directly related to pH, and I'm not sure what the geological history of pH is anyway, but I do believe that these things, again, they are minimization structures like soap bubbles. Why are soap bubbles having the shape they have? It's a surface energy, it's a surface tensional kind of thing. Surface tension is something that's very, very well appreciated in contexts that we live in. But there has been, I've been waiting for years for somebody to pick up on some of these ideas and say, okay, how does surface tension work on this kind of scale? And it's not done. And exactly what is it that, I do not believe, as I mentioned, the more complicated silicoplasmas high latitudes, simpler skeletal morphology at low latitudes, it's just a temperature thing, because people have tried to do, ratios as a predictor of a local area, but it's this area. We're always this easy relationship, and if it turns this way, if the temperature is higher than this, turns this way, and there are many variables associated with temperature in terms of the chemistry and the behavior of that water, so that is differently at a higher temperature than a lower temperature, and that's a world that we really need to learn more about. But it's a very complicated world. I mean, it gets into the chemistry and dealing with all of the and things easy. What we see at our scale doesn't necessarily relate to that scale. And if you want to talk about how these things settle and everything else, you need to look at Reynolds numbers. Reynolds numbers for a jet airplane is a whole lot different than our realm, which is a whole lot different than the realm of the microscopic. And it's quite complicated.
The high latitude here, I mean, from
they are different. They are mostly abundant when the nutrients are available, and at both equatorial areas and high latitude areas, they are more available because of upwelling. And I are working on a paper right now where we're studying some stuff, some samples from the mid latitudes, these polytropic waters, and I said, oh, hey, that's great, and I'm finding 4 silicoflagellates per slide. So the numbers are a whole lot, but that tells us something. Okay. So
microfossils that depend on nutrition condition.
All do it.
So like in a upwelling area, there must be more. And any upwelling area is obvious, isn't it?
What kind of nutrients? Which nutrients like the iron or phosphorus?
And silica. Yeah. But there's got to be other ingredients. We're always looking at the short list, and it's a much longer list than we appreciate.
I mean, we try to get it done from, or get it from Western Arabian upwelling area, near to the Oman margin.
Mm-hmm.
Can we get those? I mean,
the problem is, okay, so we started with, let's go back to this image here, back there. These 4 groups that I put up, they are the slide initially. So each one of these has its own scientific society for diatoms, freshwater salt. There's probably 1,500, 2,000 diatomists in the world. Formative for at least 1,000. Okay, several hundred for sure. Silicoflagellates are a less significant group. Their numbers are maybe a 20th of these. Okay, so if there's 1,500 diatomists, then there should be 75 silicoflagella specialists, right? You know how many silicoflagella specialists there are? And that's what I tell my students. I'm at a small university, and I tell my students that unless you're absolutely brilliant, if you're absolutely God's gift to everything, and you can be the top dog in one of these major groups. But if you're, not quite that brilliant, and realistically very few of us are, I'm not, okay, then if you want to really make a dent and make contributions, then you pick one of these little groups that people haven't done. And what I did was I made that decision, and I consciously decided I wanted to teach in a smaller school, and I probably wouldn't have they'd been accepted for the minor group at a larger school. But that smaller group, the literature fills 2 drawers in a file cabinet rather than a whole room of file cabinets. And it's allowed me to continue being a scientist and make significant discoveries in my field. And the students that tell me they want to study dinosaurs, I wanted to study dinosaurs when I was a kid. Well, with a little bit of thinking, I realized that there's 1,000 people that want to study dinosaurs and have the PhDs, and there isn't that many job opportunities for people to study dinosaurs. And the same goes for whales. Everybody wants to study whales. How many serious cetacean scientists are out there? I mean, I'm sure there's 10 times with PhDs than there are jobs and positions to study that subject. So, look for these smaller groups, because those smaller groups, they are not insignificant, okay? Yeah. Just because there's less people there does not, oftentimes the great discoveries are made in the smaller groups because we see something there that we then can relate to the bigger groups that we didn't see because the bigger groups are so big. And by the way, I'm saying about minimization optimization for zoophytes. Do you think that relates to radiolarians? Damn tootin' it does, okay? Absolutely. The mathematics would be complicated. But, I see for zoophytes, all of that relates to radiolarians. But radiolarians, which is a bigger group, hasn't seen that because it's a bigger problem.
We have one question from our scientist. Silicoplasm inform us about, can the silicoplasm inform us about climate change or during quaternary glacial interglacial episodes? They change their species variation or?
Absolutely. Changes a lot. Right now, I like the more obscure, I think. So I've been doing all my work of late in the Cretaceous Paleogene. It seems like everybody wants to study the quaternary, and it's just too large a field for me, and too much literature for me where I work and who I am to take that on. But absolutely. And one of the problems is, and let's see if anybody was going to ask me about the incorrect name of Dictyocofibula. One of the problems in Silicoflagellus is the taxonomy, and the biologists and the paleontologists are using totally different taxonomies. And the reason, I think, is the biologists don't study the literature. They don't study type specimens. The biologists call this, and this is something I got online, nice little picture, Dictyocofibula. It's a living Dictyocofibula. It's not Dictyocofibula. Dictyocofibula has been extinct since, the type specimen comes from the late Miocene, persisted a bit into the Pliocene. It's been dead for 10 million years. And yet that name has been used. That name was used in a paper in the 1930s, 1940s, and it's just continued in the biological literature. The paleontologists haven't been using that taxon since the 1970s. But the biologists are not reading the paleontological literature and asking the questions of, where's Dictyocofibula, and that is one of the problems, is that it's a group that's not studied enough. The taxonomy is complicated enough that it's been keeping new people out, I think, in some way. But it is vital. I do think that if we were, actually looking at the water column, looking at the organisms on a year-to-year basis, somebody's saying, okay, here's what we got this year. Here's what it was compared to last year. We do this in lakes and deserts and mountainsides all over the world. We have those kinds of studies. But we don't have those kind of studies in the oceans. And I think if we did that and included silicoflagellates in that, pretty soon we say, hey, what's going on with the silicoflagellates? I'm sure that that's happening. It has to be. And that would give us our first real piece of evidence, that somebody else might pay attention to. You can say, well, so-and-so is less abundant, or it's not here. Maybe it still is somewhere else. That kind of extinction kind of thing. We have insects, we have lizards, we have newts that are getting extinct, and we're not paying any attention to that. But if we have something that has a nice, pretty shape like this, and all of a sudden it just goes bonkers, that might be a visual evidence that might make people ask some questions and delegate some money.
Any other question?
I have one question, but very of a different kind. Basically, I want to know, means if you see the geological history, different forms of organisms came, either vanished or some are continuing also. So what is the basic, means what, this is the water composition that is the defining that different forms evolved at different times, or what was that main, you can...
In the micropaleontological world, micropaleontologists are fixated on biostratigraphic zonations. They're absolutely fixated on it. And for silicoflagellates, I don't think they have any value at all. I mean, there are some changes, but they don't tend to spread latitudinally by a large measure. There's not that many zones. You've got other things that do that better. But pseudoflagellates, by the nature of their shapes and their variability, they should be ideal as environmental indicators. But the problem is that we don't know exactly what environments are making these changes. And it's easy, we're looking at the six-sided versus four-sided, six-sided and highlighted. The answer is a simple ratio, and we can determine temperature. It doesn't work like that because temperature is fine, but there's all kinds of factors that are influencing each in their own way by changes in temperature, and these probably are reinforcing and interfering with each other, and we don't know that realm. And I've seen some of the isotopic studies and all these different elements and all this other kind of stuff. And that's maybe getting close to, but it's not always figure out exactly what the root cause is. I think we. And maybe there already is a lot of studies going on exact, very subtle changes amongst subtle factors associated with climate, associated with temperature. It's not something simple. It's something complicated.
No, actually, means when we are reading that the composition, they say that the oceans are in a steady state after 1 billion years of Earth history. Initial one, after 1 billion, is whatever the composition, it was more or less in a steady state. So since then, if it is in a steady state composition, so according to you, it is the temperature or some other factor there.
I did not say it's temperature. I said it's environmental variables associated with temperature. That makes it a whole lot more complicated.
Is it possible to quantify the silicoflagellates for doing the geochemistry or oxygen isotope or something like that?
Well, the skeletons themselves are quartz, and I'm not the geochemist there. There's very little. Again, I'm the silicoflagella person, and I've been very successful in getting a mathematician involved in my thoughts. But geochemists have got so many things to look at. I don't think they jump up and down with joy with the questions I present. There is so much we don't know, and when you say something little, these things are relatively simple. Diversity is not so great as diatoms or formative, but when you study something simple and start getting really confused, it tells you how much you don't know.
Yes.
And
Very true.
and there's so much. I mean, what I tell my students, I have my little climate change, as you know, all those other guys. They've heard this before from the high school teachers and everything else. What I tell them, something that I've sort of gotten from some of my nano colleagues after they drank some beer, is if you like seafood, eat as much of it as you can, because 20 years from now, it might not be there, okay? And what's going to happen if all the seafood ends? Well, that's going to, I mean, what's all the political, what's all the social, what's all the ecological ramifications of that? You know, what I'm hearing is that the pH has quite considerably in the last 30 years. And the pH changes more or less about half the amount it needs to change. Stop. Okay. And the rate of pH change is accelerating. So that's where I'm getting this 20, 25 years. Somebody can tell you more. And the nanos and the 4Ms and the other organisms, the seashells, whatever they have their larvae in the water column, it'd be different for those perhaps that are benthic. If we lose those, it'll be the KT boundary all over again. It wasn't the meteorite that killed the ammonites, okay? It might have killed the dinosaurs, but it didn't kill the ammonites.
So that means that we come back again to like KT boundaries and all these events. They are very important to study as an analog. Could be the future conditions we are facing a lot of global warming and ocean acidification.
I've not been an environmentalist. I've always kept myself restrained from that. But what's happened in the last 5, 10, 15 years, I've been seeing more of this and understanding the oceanographic implications of the dissolved CO2. It is very, very scary, and it is not being communicated at all to the populace. Again, sea level rise is important, but it is minuscule to this issue, and somehow we need to communicate that, and that is our responsibility. We need to be doing a better job of that. So that's what a silicoflagella specialist can tell you. We need to be doing better. Thank you.
Coming to the end now for this lecture, I'm thankful to the speaker, Professor Kevin McCartney, for the very informative talk. Seriously, I really enjoyed it. And all the participants who spare their time and their graceful presence. And we are thankful to the computer department to organize this talk online. And thank you very much. We will communicate further for the upcoming talks.
If anybody wants any papers from me, I know some have been sent. If anybody has any questions, email me.
Sure. I have already sent your paper to all, and I will give them your email ID too.
And
I mean, your email ID is already there in the paper.
You picked out the key papers, so you did a good choice of picking them out. Thank you. Okay, thank you.